Hi guys, you are with me. Give me 1 second. There we go. Right. Welcome. I apologize that I am a little bit late. School drop-off went a little bit not as planned as I'd hoped, which meant I needed a bit of time to get some coffee. So let me just quickly set up here and then we are good to go. Okay. Hi everyone. Hi Khadija, how are we? Hi guys, thank you so much for waiting and the fact that it is actually working with the chat, because nobody was really chatting yesterday, which was a bit saddening, but thank you. Did I get my hair cut? No, I'm getting it cut next week. I've been waiting such a long time. It's so long. Like, there we go. It's really long. Right. Okay, here we go. We are going to look at, for those of you who were in chemistry yesterday or business or sociology, I did a session on the lacoperon. I did a session on the Monday night as well. So I'm going to kind of carry these on and not go back to the other stuff. So if I've already covered it, it's in the other two lives that I did in the last couple of sessions. So just so you guys know that it is there. So please do check it out. Right, but I want to talk about biotechnology. I want to do a bit of manipulating genome today, and hopefully I will manage to get through into the immune system as well. But I've kind of dropped in where I think these things are going to be coming through. Okay, so, and also bear with me because I am using my iPad and my Mac now, so my writing is not as good, so bear with me on the whole when I do my annotations and my writing. I mean, my writing's bad as it is, but it's one of those things that I need to practice, and because I've only had it for a short amount of time, I'm not that good at it yet. And also, by the way, just so you know, one of my private students messaged me yesterday, which I'm really pleased that they did this. They messaged me to say, Christine, I have been watching one of your videos and the number you've got on the screen is different to the number I got on my calculator. Are you right or am I right? And so I redid the calculation on my calculator. And one thing that I know I do it a lot, okay, and I know I do this, is sometimes even if I read the number on my calculator, what I will then do is I sometimes write the wrong number or I'll type the wrong number. So I think it was to do with the chi-squared value, and I wrote 3771 point blah, blah, blah. Whereas actually what then happens is it should have been 3371. I put an extra 7 instead of a 3. So one thing I would say is a little bit of advice to you all is please do double check what you've written down to what's on the calculator, and do it twice on the calculator to double check that you've got the right answer. That could be the difference of a grade. And I know that my maths is my biggest struggle. So I would say, because I know that, I'm very aware of it. But when I'm doing these videos, sometimes I'm doing them a bit quick, and therefore I may have made a mistake. So please, if you do find mistakes, do let me know, because I obviously want to improve and make sure that they are not incorrect. But the key thing is, it's okay to make mistakes as long as you know how to learn from those mistakes. So, right, we're going to go on and look at the structure of the nucleotide. Now, I know that in paper 1, I've now seen paper 1, you already got something about a dinucleotide. Now, that doesn't mean they finished with DNA. It doesn't mean they finished with nucleotides. What it does mean is that they have shown you a picture and they've looked at you being able to figure out what's the same or what's different. So just so we know, whenever we're looking at a nucleotide, the only thing I'm really interested here, yes, I've got a phosphate, yes, I've got a pentose sugar, yes, I've got a nitrogenous base. I should be able to work out whether it is my purine or pyrimidine. I should be able to work out whether it's adenine, cytosine, guanine, thymine, or uracil if I'm looking at RNA. So if it's the RNA, then I need to look at whether it is a hydroxyl group here or whether it is just a hydrogen because it's a deoxyribose. So that they've kind of already covered that in the exam already. Doesn't mean it's not coming back through, but they'll be looking at a different part of it. Now, what I am wanting to focus on here is the fact that the nucleotide that I have shown you has got up here at carbon number 3. So remember your numbers, carbon number 1, carbon number 2, carbon number 3, instead of my hydroxyl group, which is what I would be expecting. And the hydroxyl group, remember, from carbon number 3, if I make carbon number 3 here, should actually be used when we are, that's my phosphate, when we're doing a condensation reaction. If I want to join the two nucleotides together to get my phosphodiester bond, then I need a hydroxyl group at carbon number 3. And that's why you can only ever bond at that point. So when the DNA polymerase or the RNA polymerase can build in a 5 to 3 direction, here's my 5th carbon, there's my 3rd carbon, therefore it can only ever add another nucleotide at the 3rd carbon of the last one. So in this case, what I'm looking at here in this nucleotide, what I'm looking at is the fact that there is no hydroxyl group there. And because there's no hydroxyl group, that is therefore telling me that what I'm looking at is a terminator base. So I'm going to talk about sequencing in a bit, but just be aware, just because they've shown you a picture in your first exam doesn't mean there's not more pictures coming. I am covering the what am I covering? I'm going to cover manipulating genomes, biotechnology in my session. If you want to jump over, Sana, to my YouTube channel, you'll be able to see my screen, which I'm displaying with the content as well. Okay, so what do we need to know whenever we are looking at the process of, if we're going to do sampling from a crime scene, if we're going to look at paternity, if we're going to do the manipulation of a genome, in the fact that we are going to genetically modify a plasmid, whatever it is that they're going to ask you, one of the key things that always comes up is about understanding of restriction endonucleases. So endonucleases obviously are enzymes. They are enzymes which are going to recognize specific DNA-based sequencing. So we know that we had, because we talked about it in yesterday's lesson, we know that we've got our non-coding regions in the telomeres, the centromeres, and the introns. So because we know we've got specific regions, we know we can use the different restriction endonucleases. And yes, that is important. Do they create sticky ends? Yes, they do. But what is a sticky end? And I think this is one of the interesting parts that you have to go back to that DNA structure to understand. So we know that we have got our nucleotide and we know that we have our nitrogenous base. So very basic drawing here. So if we have our nitrogenous base on this side, remember, because it's anti-parallel, we know we've got our nucleotides. And because we know we've got our nucleotides, we know that if I, let's hope this works. Yes, it does. Good. If I replicate that, and I'm going to do this as well. No, I didn't want that one down there. Bear with me, guys, because I'm having to relearn my shortcuts because I'm now using my Mac and iPad. So if we are going to join them together, we obviously know that we're going to have a phosphodiester bond between them, and that phosphodiester bond is between that 3rd carbon and the phosphate. So here we have our structure. Here we have our nitrogenous bases, which are going to be connected, and they are connected with our hydrogen bonds. So if I know that these are my DNA nucleotides, oh no, hold on one second, I want to add in. And if I add in an extra one, here we go. So I'm going to add this one in, and I have my nitrogenous bases. Now, so straight away I can see with my nitrogenous bases that I've got 3 hydrogen bonds. So 3 hydrogen bonds would be between my cytosine and my guanine, And if I've got two hydrogen bonds, they would be between my thymine and my adenine. So automatically, if they give you in the exam question a picture, then you should know the difference between that. But what we're looking at right now is someone asked about the sticky ends. So we know we've got our complementary base pairings. We know that we have got our adenine pairing with thymine and our cytosine pairing with guanine because of these hydrogen bonds. So if I am a restriction endonuclease and I break that bond and I break that bond, what I'm then going to do is I have then cut apart at this point here. So what I've done is I've separated at these 2 points. Now, sometimes be aware that if I am looking at this, oh, that doesn't work. If I were to move that away, that should have gone with it. Then what I have got is that is one part of my fragment. This is another part because I've cut them. And what I then have are these ends that could form complementary base pairings, those hydrogen bonds that could reform if I have the right sequence. So before I had, let's say this, oh no. Let's say that this one is adenine, this one is thymine, this one is cytosine, this one is guanine, this one is, let's say, thymine, and this one is adenine. So if I have another strand which is complementary to either these or these, then they can reform those hydrogen bonds and they can therefore form that complementary base pairs. But sometimes when we are trying to separate them, if we're using restriction endonucleases, all we're doing is we're cutting them. They could come back to the same position. So yes, we can use restriction endonucleases to break the hydrogen bonds at the specific sites, but they're not just breaking the hydrogen bonds, they're also breaking that phosphodiester bond, which was up here and down there. So the restriction endonucleases are important at breaking that phosphodiester bond, and therefore those sticky ends that we have is between the complementary base pairs. So those hydrogen bonds will remain intact and that will produce that still DNA fragment that is complementary. But what we want to do is we want to ensure that we can separate those fragments. So if we use one type of restriction endonuclease, we may, if we have this strand of DNA, we may end up with 3 fragments. If we do this with a different, so this is the same DNA strand, we're using a different restriction endonuclease, we still end up with 3 fragments, but look at the difference in the sizes. And if we were to use both of them together, that would therefore result in us getting 5 of those fragments. So DNA ligase does, yes, and to answer your question, it does form phosphodiester bonds between the fragments. So how are we able to then separate them? How are we able to, if we've used restriction endonucleases and we know they're going to cut at specific recognition sites, how can we see the different fragments? And that's where we use gel electrophoresis. So if we're going to be asked any question about this, you have to know that if we're doing a crime scene, if we're doing paternity, if we are looking at trying to find a fault in a gene, for example. So I've just seen a message that says, I saw a question about blunt ends, what are they? So a blunt end would be where the restriction endonuclease is going to cut with no sticky ends. So you are doing a blunt cut, and that blunt cut would therefore separate. So if I go back up to my example, 1 second. So if I were to do a blunt cut this time, if I do it very basic, here we go. I have my nitrogenous bases, which are going to form the complementary base pairs. And what would happen is I would form a blunt cut that would go across. There's no sticky ends. So if I do a blunt cut, there's no sticky ends, then therefore I can't then join that with, for example, a vector and those two coming together. Yes, you can identify evolutionary relationships, But when you say with manipulating the genome, be careful. The manipulation of the genome, when we talk about that as a topic area, we're talking about the genome and then how can we manipulate it. So if we're looking at evolutionary relationships, what we're looking at is how similar are the base sequences. And if we're looking at how similar the base sequences are, we can say the more similarities, the more closely related they are, therefore the less mutations that occurred. Because obviously mutations are spontaneous, and some of those mutations stay, some of those mutations are silent, some of those mutations disappear. So if we're looking at the evolution, we're talking about the accumulation of changes. So when we look at the evolutionary relationship, what we're saying is, if we're looking at cytochrome C, for example, we would be looking at the amino acid sequence, then we would be looking at the DNA sequence. Which one's better? The DNA sequence, and any changes within the DNA sequence of cytochrome C will allow us to see how closely related they are, because we can conclude that the fewer the DNA mutations there are, the closer related the species may be. If the size of the fragment is too small, would you do PCR for? Right, okay, so this is an interesting one. Okay, so PCR is really important. I'm going to bring this in a minute when I talk about, I've kind of linked that together with gene sequencing. PCR is really important because, yes, whether you're talking about just having a fragment, whether that you've isolated, whether you're talking about a whole sample, so you have taken a cheek swab, and when you take the cheek swab, what you're going to do is, obviously that's going to have your cheek cells. Your cheek cells have got the nucleus. The nucleus contains the genetic information. So we have all this information. I need to calm down. I'm getting excited. This is what I love. I love when people ask questions, because this is where you get that understanding. I am going to go through bacterial growth. It is coming up, but it'll come up in the point when I get to it, just so you know. So when we look at the PCR side of things, once I have extracted the DNA, so you've learned about extraction, you've learned about the fact that you're going to precipitate it, you need to crush the cell up if it's got a cell wall, you need to use detergent, you need to use salt, you're going to have to use freezing cold alcohol with water. So you've known about how to precipitate, extract the DNA. Then what you're going to do is you're going to amplify it. So if I've already used restriction endonucleases and I have just a small gene, or I'm going to talk about mRNA and reverse transcriptase by having complementary DNA, if I'm going to take a very small section of the DNA, if I try to do something once and it goes wrong, experiments don't work every single time. If I try and do it just once with one small sample without amplifying it, I am going to lose that sample. I am going to destroy that sample. I'm going to wreck my investigation. There's nothing I can do. We know that we would want to do a lot of repeats if we're going to do any type of investigation. We've talked about that in the past. When we do investigations, the repeatability is important. The more you repeat something, the more you are able to try to find that true value. So if I'm taking a small sample, whether that is I have a sample of DNA, whether that is I have a fragment, whatever it is, I need to amplify it so that I can get as much as possible. So yes, we would always talk about the amplification part of it, and I will bring that in a minute. But one of the things that they love to throw in there is the understanding of what's going on. So how did I get that fragment? If I'm going to have this small fragment, where did it come from? Restriction endonucleases. If I'm going to do genetic modification, what do I need? Restriction endonucleases. So restriction endonucleases come in all the time, so it is important to know the difference. With the gel electrophoresis, the key thing that you need to know is, obviously you have your loading wells. Because you have your loading wells, this is where you're going to place your DNA sample that you have taken, that you have amplified. Now, I have done this a number of times with my students. It depends, A level 25, it depends with regards to what you're looking at. If you're just looking at the cells being, you're extracting the DNA from the cell, you may want to amplify that fragment, amplify that DNA sample, and then in the next sample, then you're going to use your restriction endonucleases. So it really does, it depends. They're not going to penalize you, I don't think. Now, I could be wrong on this because there is lots of interesting things out there, but I don't think that they will penalize you whether you say restriction endonucleases before PCR, if you say PCR before restriction endonucleases, unless they have given you actual information in the stem of the question and they've gone step by step by step by step by step. So it depends on what they've told you. If you have isolated the gene from mRNA, the restriction endonucleases are needed for the plasmid, and so therefore you wouldn't need it for the gene. But if you're trying to isolate the gene, then yes, you would need the restriction endonucleases first. So it's really just dependent on how they ask it in the question. So don't get hung up on I must have this order. As long as you've read the question and you've put it into a logical order, then it's important. And if they're going to ask a question, which is as they've done in the past, where they've said A, B, C, D, E, and they've given you a definition of each of the different things, then if they do that and they want you to put the A, B, C, D, E in the correct logical order, there will be a very obvious order for you to be able to do, not an ambiguous one. So just be aware of that. Okay, so going back to this then, if we're looking at the agarose gel and we are going to place our DNA sample in this loading well, you need to make sure that the loading well is positioned in the right way. And the reason being is because students have done this in the past where they did it the wrong way. If you don't put it in the right way and you don't remember that DNA has got a, so we know that DNA has got a negative charge because of our phosphate group. Therefore, we need to put the loading wells at the side that we're going to put the cathode. So when I've done this with students in the past, they actually put it the wrong way around and it resulted in us getting no results. Now, if that was the only sample we had, we then couldn't repeat the experiment. But by doing PCR, by amplifying the fragments that we had, by having lots of copies of it because of semi-conservative replication, so this is where they could bring in your DNA replication knowledge and PCR together because of what's going on, or they could link it, as we're going to talk about, to the sequencing part of it. But by amplifying my DNA means I can do this in lots of different times. So it means I can repeat the experiment. What we're going to have then is, we know that we're going to have our DNA fragments which are going to move towards the positive electrode. They are going to be moving away from the negative electrode. And when they do that, they're going to move through based on the fragment size. Okay, so the fragment size means that the smaller they are, the further they will go. So if I go back a second, this is an actual gel electrophoresis. My students and I, a number of years ago, used to be part of the Amgen project, which is a biotechnology company that connected with the Cambridge University. And so I would go and do the course every year, and my technicians would do the course every year, and we would then get a loan of their equipment, and we would then actually transform bacteria. We would use restriction endonucleases, we would use ligase, we would use gel electrophoresis, we would use fluorescent markers so that we could visualize. So what we have here right now in my picture is we actually have this really long one. So this is group A and group B. So we had two groups that were doing this one. This was the best one, although it still wasn't very good. So this is A, this is B. And what we had is each group had their own ladder. And that ladder is used because that ladder shows you known fragment lengths. And so therefore we could see whether or not our DNA had been successfully recombined, had been successfully separated with restriction endonucleases. We could see that based on the position that they actually were going. Doesn't necessarily mean, sorry, just to answer the question here, user 765. Stuff from module 2 that came up in paper 1 won't come up in paper 2, right? OCRA, by the way. It doesn't necessarily mean that that's true, because you may think that DNA is not coming up, but DNA is so important in paper 2 that they can't not come up. That's why module 2 is 1, 2, and 3 for the papers. Paper 1, paper 2, paper 3 is module 1 and 2 in any paper, and it just depends. They may take a spin on it, so rather than them looking at the nucleotide structure as they did in paper 1, what they might do now is look at your understanding of DNA replication, which is nucleic acid nucleotide topic. So it just really depends. Don't assume, this is one of the things, don't assume anything. It's why I am hesitant to make any predictive papers, because I don't know what they're going to do. I have strong gut feelings where they're going to go, but I don't know. Okay, so making sure I've answered all the questions so far. I am going to bring in the reverse transcriptase. That's still coming. The other thing to note, they asked in the past about a buffer solution. Now, obviously, we've got DNA that is going to need to move, and it moves through the agarose gel. And so therefore, we need to ensure that the buffer solution allows the flow, because we're using a cathode and an anode, we need to allow the flow of the ions through, and we also need to allow the flow, therefore, of the DNA. But we need the buffer solution, because the buffer solution is obviously going to be used to maintain the pH. So yes, we need it for pH to control the pH of the solution, because pH can have an effect on the DNA fragments, but also we need to use it so that we have a liquid, a solution that allows that movement of the fragments through the agarose gel, also allowing the flow of electricity, which is why it is electrophoresis. Now, the DNA ladder, one thing to note when we're looking at the DNA ladder is the fact that you have got your different fragments, and obviously they did that in last year's exam. That doesn't mean they're not going to bring it in again. It may be more them looking at your understanding of why would we use a DNA ladder. So a DNA ladder is like a using of a calibration curve when you do a colorimeter. It's a way in which you have known samples, known DNA fragment sizes, so that you can then compare against those known fragment sizes. So that's important to note, that part. So now let's go back and look at our PCR, because everyone's been talking about it and wondering about the PCR side of things. So whenever we do DNA sequencing, or whenever we are going to amplify, if we were doing a paternity test, if we were trying to amplify any small sample, we are going to use PCR, polymerase chain reaction. So what it is it's a thermal cycler machine. So it's a really small machine. It's really fascinating. So we had to use this. It's a really small machine. And what you do is you've got your DNA sample, and that DNA sample has been provided with key things. What do we need? Well, we need free nucleotides. We need the DNA sample, we need free nucleotides, we need there to be primers, and we also need there to be an enzyme polymerase, Taq DNA polymerase, to build the new strand. Because we already know from my previous sessions that I've done, so obviously the topic areas that I did in the paper 1 prep could have come up. If they haven't come up, like semi-conservative replication, they can come up here. So we know that if I have my DNA fragment, I know that those DNA fragments are going to be separated. And if they're going to be separated, they are therefore going to then have a primer that starts off, and then they're going to have the extension of the chain. And so what we get is a semi conservative replication. I start with 1, I've now got 2. So to do that, the first thing we have to do is break the hydrogen bonds. Well, obviously inside our cells, we've got helicase. Well, we don't want to add an extra enzyme in here. We don't want anything else to contaminate our sample. So what we're going to do is we're going to have it where we're going to denature it. We're going to use temperature. So what's happening? We're increasing the temperature. When we increase the temperature, we're going to increase the kinetic energy. When we increase the kinetic energy, that is going to denature. It's going to break the weakest bond, the hydrogen bond, the intermolecular forces between the nitrogenous bases. So by denaturation, by 96 degrees C, by raising it up, we are going to break those hydrogen bonds. Breaking those hydrogen bonds, we're then going to do what's known as annealing. And to anneal, we have a primer, which is going to be complementary to the fragment, because DNA polymerase cannot build from nothing. Now, we don't talk about it in OCR, about the fact that you need a primer until this point here. But actually, there are other enzymes involved in DNA replication. So if you go on and study this later on, if you go to university, you're going to turn around and be like, Christine, you told me a lie. You said it was this. And it's like this, and this. And that's because at the level that we're at level 3, we have to, and that's what you're doing right now at a level 3 qualification, we have to try and make it correct, but to a level that your brain can cope with. And even that, OCR have so much content in there. So if you do go on and study this more, you're going to learn more about the fact there's DNA polymerase 3, the fact there's DNA polymerase 1, and they have different roles to play. The fact that you have primers that are going to be joined and then need to be removed. So what you have to understand for this, for your exam, is that it always follows the same thing. Denaturation, then annealing for the primer to join, and then we're going to extend, and the extending is going to be done through the use of Taq DNA polymerase. So we have the Taq DNA polymerase. Why are we using Taq? We're using Thermicus aquaticus. We're using the enzyme coming from a thermophilic bacterium, so a prokaryotic organism that has got a production of a protein. How did it produce the protein? 70S ribosome, protein synthesis, and that optimum is at a higher temperature than what our cells need. So if you understand that, again, I've given you 96 degrees, I've given you 50 degrees, I've given you 72 degrees. See, it depends on the question that they give you and what range they've decided to give you for that. But one of the key things we would want to be talking about is that we want the temperatures to be at the optimum for the process to happen. So what's going to happen? It's going to do this. Denaturation, annealing, extending. Denaturation, annealing, extending. So when it does the next step, what we're going to have is, instead of, hold on one second, this now, what we're going to do is that is going to now, let me move that. Oh, no, don't do that. Did that wrong. Now what we're going to do is we're going to do the second time of doing this. Seriously, you guys are so good at using your iPads. You make it look so easy, and I'm having to relearn because I haven't done this in such a long time. So now, because we're separating them, we're denaturing them. Now we're going to have our primer come in. I'm making this very basic, by the way. And then we're going to have our new strand be built. And that's going to happen each time. So it's a cyclic thing that's going to happen. And how many generations is it going to do that for? So if I start with one DNA sample and I do, let's say I do this over 2 hours, and in 2 hours I can do 30 cycles. Therefore, I would have 2 to the power of 30. So the number of DNA fragments that I would have would be 1 times 2 to the power of 30, which, oh gosh, now I don't know where the calculator is on this one. Calculator, there we go. Oh, go away, give me scientific. Where's scientific? There we go, much better. Right, here we go. Can tell I'm doing this live. So 1 times 2 to the power of 30. So DNA fragments, what would I have? I would have 1. Oh, hold on. That would equal to 1073741 So do make sure that you are thank you Waterpin, that's awesome. Yeah, make sure you are double checking. As I said before, sometimes I make a mistake, so I would double check what I've written with what I have actually got there. I've got hair stuck to me. Okay, so that is something they could ask you. But what we're interested in is, I'm going to look at DNA sequencing. So the PCR is always the same. It's the same process. We're going to denature, break the hydrogen bonds. We're going to anneal by having a primer. We're going to extend, and we're going to have Taq DNA polymerase. Oh, actually, before I move on to that, one of the questions they could ask you could be about why, if you were leaving it for longer, let's say I left it for longer, why would I not get more than, let's say, this number of DNA? And so if that comes up, that's them looking at you saying, well, we may have run out of nucleotides. We may have run out of primers. Even though we can keep the cycles going, we can keep it going for longer if we don't have any of the free nucleotides. If we don't have them available, what's going to happen is, yes, it would denature, it would separate the strands, but they just join back together again. And so it's trying to think outside of the box, trying to think about where might they go, because that would be assessing your understanding that a PCR is about amplifying, but we can only amplify up to a point based on the free nucleotides that we've provided it with. Okay, so if I'm going to do DNA sequencing, then what I need to understand is that terminator base that I had before, that terminator base is going to terminate the process of replication, is going to terminate at random points. Now, obviously, if I'm doing it many cycles, in this case, I said there was going to be 30 cycles, all of the possible DNA chains will be made because they will have inserted themselves, these terminator bases, at specific points. Now, the DNA will then be separated according to its length, like we looked at with gel electrophoresis. It's OCR for those of you who are just joining, but it doesn't mean it's not relevant, but yeah, it's OCR. So the DNA fragments are going to be separated just like they were in gel electrophoresis by that electrical field. So we've got an electric field applied to a capillary of gel. We know the DNA is negatively charged. We know that it's going to migrate towards the positive electrode through the gel, and we know that the smaller fragments are going to move faster. Now, be aware, they haven't done it in a while. They've done a lot of TLC lately in the last couple of exams, so they could actually ask you to compare the difference between TLC and against using gel electrophoresis. So that would be a way in which they're looking at you understand the difference between the two. This one is separating it due to the fragment size, whereas obviously TLC is about the solubility and the interactions. Do you have to say Taq DNA polymerase, or can you just say Taq? You should always try and make sure that you're saying things like DNA polymerase, because we're talking about us doing DNA replication. So the more you get into habit of saying DNA replication, DNA polymerase for DNA replication and RNA polymerase for transcription, the more you are likely to ensure you're not penalized if they decide, actually, we're going to be specific, we want DNA. So I would always say those kind of wordings should be a go-to, just standard. Okay, because polymerase just means an enzyme that's going to make a polymer. So is it a polymer of RNA nucleotides, or is it a polymer of DNA nucleotides? And that's where the distinguishing part is. And that's why they'll be wanting that specificity. So if we're doing, we're 1 times 3. Okay, Imogen, come back to your one. So I'm saying if I start, because that's a good question and it is one that they could bring up. If I had one DNA fragment to start with, right, we know that that one DNA fragment in the first point of being replicated is going to give me 2. I'm going to try and draw this really, really small. Now, if I do that again, I know that that is going to now give me 4 fragments as I showed above. 1, 2, and then I need my blacks. 2, 2, 2, 2, 2, 2. Yeah, and if I did it again, I would get 8. If I did it again, I get 16. So there would be number, so that's replication number 1. There's replication number 2. I'm going to keep doing this, and I'm going to do it to 30 times. So what I've done is I started off with 1 and I have replicated. I have done denaturation, annealing, extending 30 times. So because I've done that 30 times, I have doubled it 30 times. So 2, because I'm doubling it each time, to the power of 30. If I start with 1, it was 1 times 2 to the power of 30. Now let's say instead of having just 1 fragment, I have got 5 fragments. I would therefore have 5 times 2 to the power of 30. Which, if anyone wants to do that and give me the answer, that would be where you would get your answer for the next one. No, it was just an example. So I would say it would never normally go past the 30 cycles. So it's normally, I mean, I'm saying normally, we're talking a good, what? My eldest is 7 now, so I haven't done this in 7 years. So last time we did it was about 2 hours it took, and that was enough time for it to do 30 cycles. But it'll probably be much faster now. So it really just depends what they give you in the question. So I could change that. Instead of saying 30, I could say we do it for 18 cycles. And if I do it for 18 cycles, thank you, Watchpen, then I would do 5 times 2 to the power of 18. So however many times they tell you, that's what you are doing. The Sanger sequencing, so someone's just asked about Sanger sequencing, that's what I'm covering now. So the Sanger sequencing is the same sort of thing. The only difference is now what we've got is as the DNA fragment moves through, so this one obviously, hold on, let me see if I can do this. This is where my nice little diagrams. So here is my DNA fragment, which is the shortest one. That one went through the detector first, and that one had a fluorescent dye which indicated that that was a cytosine nitrogenous base. Naomi, to ask you on that one, would you be given a time frame? I think if they're going to give you a time frame, that would be if you were looking at population growth curve, which I think could potentially come in again. So that's what I would say if they're going to give you that one. With regards to the primers are short fragments of known sequencing, and they are needed to bind to the DNA fragment to start with, because DNA polymerase cannot bind without anything else. So here we have, that one was C, then we have T, thymine. Then if we zoom in again, we can see there's a green one, so that's my adenine, and we can see that we have got another one, which is my cytosine. Now the next one that's going to come through is guanine. So when that makes its way through, once it is past the laser, it will give a fluorescence off, and that fluorescence will be detected, and that will allow us to visualize the sequence. So this would be on a computer screen, by the way. So if they give it to you, so they could do it where it goes like that, and they've missed out some of the letters, and you're meant to figure that one out if they decide to do that. It really just depends. So is the Sanger, so the Sanger sequence is the way in which Sanger did it. He established it to begin with, and then now what we use is we use the high throughput sequencing. We basically we use the advancement of technology to do things at a faster rate. Do you need to know about dideoxynucleotide triphosphates? No, you don't. You used to have to know if you're doing a different exam board like IB or anything like that, then yes, you do. Although I think they've removed that now from there. But no, you don't need to know. Although they have given it to you in a question, they're not expecting you to know this content. They're expecting you to apply your knowledge to the content. So if you know that you have got a triphosphate, you know that that has got 3 phosphates, and just like ATP, that has lots of energy, and that energy through the process of hydrolysis by breaking away the other extra phosphates, that provides the energy for the joining together of the bonds. So if you did chemistry, you obviously know that you need energy to break bonds, but you also need energy to make bonds, and therefore it's about what they're expecting you to know in a question about that. That would be more them looking at your application skills on the knowledge that you should already know. So you'd be given something in the question on that. Okay, so why would we be using DNA sequencing? Why would we want to know about the different DNA bases? Because we could be looking at whether it's to do with has a mutation occurred. So is there a genetic disorder that's happened, and the genetic disorder is causing a problem in the population? If it's causing a problem in the population, what we're going to do is we are going to try to find a way to cure it. We're going to try to find a way to help reduce symptoms. We're going to try to find a way to alleviate the issues that it has for the human population. So if that's the case, by sequencing the DNA, by comparing a disease cell compared to a non-disease cell, by comparing a extinct species DNA to a living species, all of this by sequencing means we are going to be able to find ways to help our growing population, to find ways to increase the diversity that's out there. Is the Sanger sequencing using fluorescent dyes? So they are fluorescent DNA probes, slightly different. So the nitrogenous, let me get this right. The terminator bases will have dyes associated with them, and those dyes are the ones which are going to be given off the fluorescence which will be detected. So it's a way in which we can visualize, and using the technology now to be able to see. So if we're not using fluorescent dyes, we're using DNA probes. Be sure at what it is they're actually asking in the question. So make sure you use the information this time of the question to help you answer the question. So this is the thing, it depends what the person who's written, or the people who've written the exam questions, what they want to assess your knowledge on. As to what they're going to actually give you in the question and what they want you to pull forward. So don't get hung up on knowing exactly where to bring it in. Michelle, Jill, it's really interesting that you've just said that, because that just came into my mind that, oh crumbs, hold on one second, that I haven't actually mentioned the southern blotting part. Why are you not working? There we go. Right. And I did just think that through when I was talking about whether we're using fluorescent dye or whether we're using the DNA probes. So if we have amplified, right, and if we have separated, so let me just bring my picture down here. Get rid of that. Get rid of that. I'll put that there. There we go. So if we're looking at my DNA ladder, I was talking about this with one of my students yesterday. So I've got my DNA ladder. I've got my separated fragments. At the point that we're at right now, we wouldn't be able to visualize them. So if I've got my fragments, if I don't do something, I wouldn't be able to see them. So what we have to do, we know that our DNA is double-stranded. So what we do is we know that our DNA is double-stranded, and because we know that our DNA is double-stranded, what we need to understand is to do southern blotting, the first thing we're going to do is we're actually going to use an alkaline solution that is going to make it single-stranded. So if it is single-stranded, what we can then do is we can then actually take that and we can transfer that through the process of southern blotting onto a nylon membrane. And what we do is with the nylon membrane, once we have actually joined it together, what we're then going to do is if we move that away, we can then use our DNA probes, and our DNA probes are going to be complementary, and they're therefore going to have a way in which allows us to visualize. So by doing southern blotting, what we're doing is we are taking our fragments. Now, this is going to happen with all of them. So basically, we take our agarose gel, we immerse it into an alkali solution, and that therefore will give us our single-stranded fragment. And then we want to do southern blotting where we remove it through capillary action, going to get that onto a nylon membrane, and then we're going to use a DNA probe, and that DNA probe will do complementary base pairing with that strand if there is complementary base sequence, and that will therefore allow us to visualize the presence of, for example, a sequence that we're looking for that could cause a disease, let's say. So you asked about what is a satellite DNA. So variable number tandem repeats, VNTRs. So variable number tandem repeats. So let's say I have got a ACG. Oh, crumbs. That was awful. Give me 1 second. I might need to. Let's try that again. Slow myself down. C, G, A. So let's say we've got ACGA. I'm just making this up, by the way, on the spot. So I've got ACGA. Now, if I have a variable number tandem repeats, then what would happen is, let's say we've got this 4. Now, actually, it would be more of a microsatellite than a mini satellite if it's 4. But I've got ACGA. Then I'd have ACGA, ACGA, ACGA. It does not like the way I write C's. ACGA. So here we have, there's number 1, there's number 2, there's number 3, there's number 4, there's number 5. So here we can see 1, 2, 3, 4, 5. So I have 5 repeats of ACGA. Now, let's say, micro would be 20 to 40 bases. Would micro be 20 to 40 or is it mini? Micro is smaller than the mini. You sure it's not the mini satellites on that one? Anyway, doesn't really matter on this one. They're not expecting you to know it. It'll be a case of them giving you the example in the question. If I now have, instead of having 5 repeats, I have got, there we go. I did wonder because I was thinking the same thing. I now have 15 of those repeats. So it's still ACGA, but it's 15 Thank you. This was actually a picture I took last Tuesday, and this is really fascinating as well. And I want to do a video on it, but I've not done it yet. With regards to the tree, it was massive, it was huge, and they cut it down, and now the new growth has started, which is really cool. I should put a combination of that one. So yeah, so does that make sense with the variable number tandem repeat? So if we actually know the sequence, because we've done DNA sequencing, and then we use the fact that we know how there can be different number tandem repeats, it means we can see if someone is, for example, heterozygous for something or homozygous for something, because we can look at how the bands separate out, which is obviously what they looked at in last year's exam. Now, Naomi, you asked about computational biology. So what is computational biology? We've got to realize that we have computational biology and bioinformatics. Now, these have been huge over the last couple of years in the assessment side of things. And the reason they've been huge in the assessment side of things is because we've had so much advancement. Now, Alan is a computer scientist. He's a engineer. He does a lot and has done a lot with genomics. So what is bioinformatics? Bioinformatics is the building of the software. It's the building of the programs to store the data. So building the databases. If we are going to do DNA sequencing, if we are looking at the sequences of a gene and we want to compare, for example, I gave that a diseased and a healthy cells sequencing. If we want to compare the differences, we're going to want to store that information somewhere. That is what bioinformatics is, where we are taking the data, the amino acid sequence, the proteomes, the DNA sequencing. We're taking all this information and we are storing it in databases, and we are therefore using algorithms, simulators, statistical analysis to then model and predict what's going to happen. So is it a case of the misfolding proteins causing potentially Alzheimer's or dementia? And if we can find a way in which we can figure out what combination they come together, how is that going to then be useful to help others later on who may develop the diseases? So what you're doing right now, you guys, by studying this information, you could be the next generation that goes on to find a cure for a disease, because you have learned about complementary base pairing. You've learned about DNA sequencing. You've learned about how it all comes together. So it's important that you look at, going back to the question, I could talk about this forever, but if you go back to the question that they give you in the exam, because we don't know what they're going to give us, go back to that question, have a look at that question and see what information, what little breadcrumbs are in there for you to be able to pull out of the question. When it comes to synthetic biology, if I synthesize something, I am making it. So if I were looking at synthetic biology, we could know that we have got a DNA sequence, and that DNA sequence is going to cause a disease, let's say, right? So what I want to do is I want to synthesize a sequence that's slightly different, and then I'm going to insert that into a patient that is potentially suffering from a disease that could actually help. So it's that whole thing of how are they going to use this information? How can they then improve what they're doing? What's the difference between... What do you mean by IO technology and computational biology? What do you mean by that? I'm not entirely sure what you're meaning there. Also, does it matter if we use VNTRs or STRs? I don't think it's going to matter unless they've given you something in the question about that, because there's no expectation of you knowing the variable number tandem repeats or the short number tandem repeats. So I don't think it's going to matter too much in the exam. It depends on the question that they give you. That's important for that. Sorry. Sorry, but I haven't heard of this nylon thing. That's part of, if you do OCRA, then you should be looking in your textbook. It's on the page about DNA sequencing. It's a different colored box in the page as well. So do make sure you are looking in case I am going to do biotechnology. Oh, that's what you were trying to say, biotechnology. So what's the difference between biotechnology and computation? So biotechnology is completely different. Biotechnology is where we're using living organisms to produce something that we want. And I'm going to talk about biotechnology in a bit. So with regards to computational biology, this is about simulation. This is about mathematical modeling. This is about using the information that we've stored to then create these models of what we think is going to happen so that we can then look at how drugs may be responded to. Whereas biotechnology is actually using living organisms to produce the products that we want, in a sense. Okay, let me just finish this last bit on phylogeny then. So obviously, module 4, you need to know about classification and evolution. And I've got a feeling that classification is coming through a bit more this year. Although they did the thylacine question a couple of years ago, they didn't really look at it in the way I could see it coming forward. So, do you know about phylogeny? Do you know that this is the evolutionary relationship between organisms and it's the basis for our classification? So if they are put into the taxon, so you should know about your taxons, which is your, let me try that again. No, there we go. Kingdom, phylum, class, order, family, genus, species. KPCOFGS. If you haven't seen the classification rap video, then you are missing out. It is so cringeworthy, but also so awesome. When I was in the class, I would actually sing the song. The students would be like, "Oh, Christine, stop." But it is something, when you find something that works, that sticks in your head, that is going to help you. So kingdom, phylum, class, order, family, genus, species, KPCOFGS. So yes, you can do the KPCOFGS really quickly, but can you actually relate that back to kingdom, phylum, class, order, family, genus, species? So if we have organisms that have been classified within the same kingdom, Animalia, Protoctista, fungi, plantae, prokaryote, if they have been classified within the kingdom, they have an evolutionary relationship, okay? If they are within the phylum, are they within the chordata? Do they have a chordae? Are they within the subphylum Vertebrata? Are they within the class? Are they mammalia? Now, you don't need to know these, although I am giving them to you. They will give it to you in a question. So if they share the group, if they're in the same group, therefore that is going to mean that they are classified because they have an evolutionary relationship up to that point. So their DNA may be similar for whatever it is they're looking at. If it's cytochrome C, if it's hemoglobin, blah blah blah, whatever they decide to give you. Obviously, we've got the common ones we use, but they always throw in so this protein, blah blah blah, and students go, my teacher never taught me that. They taught me cytochrome C. Doesn't matter what they give you as the example. What matters is about how you're applying your knowledge. I am going to come into Khadija, I'm going to bring in the domain systems in a minute. That is coming. So if we are looking at this, if we're looking at which of the groups that we are putting them into, and we are remembering any time we do this, we are always going to have our genus and our species, which is the way in which we use our binomial naming system. That's how we name our organisms. So it could be that we have, for example, a common ancestor. As you can see here, we've got an African common ancestor. And what we can see is this is time. Be aware, they may give you an arrow which represents time. They may not. and that will have a big thing of you wanting to talk about that if they give it to you. So if we are going to represent this evolutionary relationship, we normally do it in what's known as a phylogenetic tree, which is what you can see here. They may give you one. I don't know why. I thought they were going to give you one last year and they didn't, so it makes me think that it's potentially coming this year. Those branches are showing how different species have evolved from a common ancestor. So we can see quite clearly that we have, we have our African common ancestor, and then we have a divergent part. We haven't done the rest of module 4. No. So I'm coming back to module 4. I'm doing things in the order of where I think and how I think they're coming through in your paper. And so if we have a African common ancestor and we have a divergent point that we can see here, the branches are showing how these different species have evolved. Now, they may ask you about speciation in a question, and I'll talk about that if I have time a bit later on, or they may just look at the evolutionary side of things. So how do we classify? We classify based on embryological evidence. We look at the early stages. So you obviously know about your Hox genes, you know about your homeobox and how the body development. So looking at that embryological evidence of how does the early stage of an organism develop. Now, they did quite a lot on that last year, so I'm not sure if that's coming through. Doesn't mean you shouldn't revise it. I just don't know how strongly they are going to bring that through. We have behavioral evidence. So do they have behavior and social organizations of the organisms? Is that important? So for those of you who've been in one of my lives before, you'll have heard about me mentioning that I went off to Gamboa in 2014. And whilst I was in Gamboa, which is close to Panama Canal, when I was in Gamboa, I got the opportunity to go to Panama and to go to the Smithsonian University and go to a lecture. And one of the lectures was on the fish and the personalities in fish, and I was like, oh my gosh, this is crazy. People are spending lots of time studying this, but it is actually really important understanding how behavioral evidence can be used and that social organization to allow us to see how these behaviors have actually evolved from common ancestors. So if we're looking at the hominidae, when I was at university and I was studying my evolution and I was looking at the evolution of the Australopithecines, the Homo habilis, Homo erectus, Homo neanderthalensis, Homo sapiens, when I was looking at the evolution, the different behaviors, the Homo erectus upright, the Homo habilis having the tools, I was doing that studying based on the information. Were they hunters? Were they gatherers? What were they? So by studying the organization, the behavior of the organisms, we can see why there may be that there are species that have to be classified as different species because they have different behaviors and they no longer mate, and therefore because they no longer mate, they have been reproductively isolated and then they have evolved. So if it's sympatric speciation or if it's allopatric speciation, depending on if it's geographical or whether it is within the same environment. So we have to use all this information together. Now, obviously with advancement, molecular evidence, that DNA sequencing has improved the way in which we have classified organisms, but it's also meant that we've had to reclassify organisms. And this is where I feel like they could go with a question. They could talk about the fact that they classified these organisms in the past by using one of these methods, and now we're reclassifying them, or maybe we can't reclassify them. Why are we reclassifying them, or why can't we reclassify them? So just something to think about when you are looking at that content. Can you explain the phenological definition of a species? So the definition of a species is about the individuals within a population in a given place and time being able to reproduce and produce fertile offspring. So when we're looking at whether it's a species or not, we're looking at, Are they able to produce fertile offspring if we were to have them mate? Then we would start to look at their genetic information. Are their DNA sequences the same or are there differences? So when we look at that base sequence, we can talk about if there are few mutations, we would say that they only diverged relatively recently. So obviously, the more time that has passed, so if you look at this, for example, we can see that time in the arrows. We've got our time in our arrow. Therefore, there was a long time ago since they diverged, whereas C2 and S1, these two have diverged more recently. So they share a more common ancestor. So the sequence differences that accumulate can accumulate gradually, or if you're doing another example, you'll learn about the gradual or punctuated changes. The key thing is there have been changes that have happened, these random mutations, this accumulation of the sequence differences, and that is why it's about that evolutionary relationship, because evolution is the accumulation of the changes in the allele frequency. So we know that these sequence differences are going to accumulate gradually, and therefore we know there is a positive correlation between the number of differences between two species and the time since they diverged from a common ancestor. But remember, because they love to throw this one in there, correlation does not mean causation. And so we have to know the difference between whether it is a change that has happened, which is that they have diverged from a common ancestor, or whether it was a convergent evolution where they have the same structures and functions, but they were from different for different reasons because of the niches that they live in. Okay, guys, I'm going to pause for a minute because I've been going for 71 minutes. I will be back. Bear with me. Please do bear with me. Oh, that's actually there now. Oh, nice. Okay, that's quite nice because it means I can now get rid of that. Perfect. Yeah, bear with me 1 second. I will be back, guys. I appreciate you all staying with me for this. Okay. Oh, I don't know what was going. Oh, thank you. Someone just said I'm very smart. It's more a case of that I've done this course so many times that I know the course, and therefore I am just trying my best to keep you guys to a point where you are able to go away, you're now being annoying. 1 second. I did something and now it's like, no, I don't like you. Oh, give me 1 second, guys. Okay. Was PAGS experiments must we follow for paper 2 for OCRA? So this is an interesting one in that it's not a case of that you have to follow the specific PAGs. It's about what they decide that they're going to ask you. And so I would say try your best to not worry too much about that and more about knowing the content, because they can ask anything. What uni did you go to? So the uni I went to for my undergrad was Dundee. The uni I went to for my postgrad and masters was Cambridge University. It's why I'm in Cambridge. One of the things that they always ask as a question is about the fact that classification, the link between classification and phylogeny. Classification is based on phylogeny. So if we're going to classify organisms into the kingdom, phylum, class, order, family, genus, species, we therefore need to look at those evolutionary relationships so that we can connect the two together. Give me 1 second, guys. There we go. Right. Okay. So looking at the domain system then. So obviously we've talked about classification. We've talked about the phylogeny. What we need to understand is it doesn't just stop at the kingdom. We have to go up to the domain. We have to understand that because of the advancement of science, because we know about all this information, because we can do DNA sequencing, because we know about the diversity in the world, we need to understand that sometimes we get it wrong. Scientists make mistakes. Scientists learn new things and they adapt and they change based on what they are learning. So in 1990, obviously, Carl Woese used his results. So he started studying cells. He was studying them and he was looking at ribosomal RNA. He was looking at cell membranes. He was looking at the lipid structure. He was looking at antibiotic effects. And what he then did is he reorganized into what we now use, which is the 3-domain system. So we have eukarya. So any of the kingdoms that fall into the eukarya domain have membrane-bound organelles. They have a nucleus that contains their DNA, which is linear, that is associated with histone proteins. They have got the ATS ribosomes. So these sorts of structures that we use to distinguish organisms helps us to identify which of those groups we should put them into. So when we look at, for example, their RNA polymerase, because we know RNA polymerase is needed for transcription. Transcription is needed to copy the genetic code so it can leave the nucleus. They have got lots of proteins involved in their RNA polymerase. Now, the 4 kingdoms I just talked about, the Protoctista, Animalia, Plantae, and Fungi, you should know if they do ask you a question about classification, you should know how to classify them, and you should know about, are they unicellular, are they multicellular, do they have a cell wall, do they have a cell wall made up of cellulose, do they have a cell wall made up of chitin, do they have a cell wall which is made up of murin, or do they have a cell wall, in this case with Protoctista, if they do have a cell wall, is it made up of cellulose or is it made up of a silica? It really just depends in the question. And they've done a question in the past where they gave you an organism that had some structures that were the same, some structures that were different, and asked you to then classify how it is one of them, and you have to look specifically at what they've told you. So if it were, for example, it's got a cellulose cell wall, we can automatically remove fungi and animalia. So we can classify it as not being those. We've eliminated those because it has a cell wall made up of cellulose. Is it an autotroph or is it a heterotroph? So we know that plants are autotrophs. So therefore, if we're told that they are only autotrophic, they only produce their own organic compounds through the use of in plants using light, that would mean that it would be plantae. But if they told us it was unicellular, then it can't be plants, it has to be Protoctista. So they're going to give you, I think, something along the lines where you're having to use this information and decipher which of them that we are going to put them into. So if it is the eukarya domain, we know it incorporates those four kingdoms. If we're looking at bacteria, yes, we know prokaryotes have 70S ribosomes. We know that their cell wall is made of peptidoglycan or it's made up of murin, depends on which one they decide to go. Prokaryote is a kingdom, yes. And what they realize is they used to have the 5 kingdoms. So it was prokaryote, Protoctista, Animalia, fungi, and plantae, so 5 kingdoms. And then what they said is, oh, hold on a minute. We have got eubacteria, true bacteria, but we also have archaeobacteria. So they both have 70S ribosomes. The cell wall is slightly different. The RNA polymerases are slightly different. And therefore, because of that, we need to separate them out into a 6 kingdom rather than 5. So if it's archaeobacteria, if it's eubacteria, therefore 6 kingdoms rather than the 5 kingdom where we grouped all the bacteria together. Do we need to only remember ATS? Oh, what about membrane? Yes, membrane bound organelles is really important for the eukarya. So the ATS ribosomes is my way of distinguishing evidence that they tend to talk about. So when it was Carl Woese, he was looking at these ribosomal RNA. He was looking at the cell membrane lipid structure and he was looking at the antibiotic effect. So that's what I was showing here. It really depends what they're looking for. But if you are talking about how to distinguish a prokaryote from a eukaryote, then you should have your list. Linear chromosomes, DNA associated with histone proteins, membrane-bound organelles, ATS ribosomes, they're your go-to answers for that type of question. Right. Okay, here we go. We are now going to switch over slightly, but not quite, but we're going to focus on our prokaryotes and we're going to focus on biotechnology. Will I be going over the nitrogen cycle at some point? If I can't do it in this session, which I'm probably not going to have enough time to do it today because I've obviously got my own clients later. What I will do is I'm going to go live tomorrow again, tomorrow night, not tomorrow during the day, because I've got basically all my students, if they're not doing chemistry or maths, I'm seeing them all tomorrow. So I will go live tomorrow night after I've seen my last private client. So anything I haven't covered, I will cover there. But I have done quite a lot on the nitrogen cycle in my lives in the past, which is why I've not started with those right now. So if you look at some of my lives that I was doing on a Monday and a Wednesday previously, you'll see a lot on the nitrogen cycle. But I do feel like if they are going to bring in the nitrogen cycle, it may be about the bacteria and the prokaryotes. Potentially, like, how do we actually classify Nitrosomonas and Nitrobacter? How have we classified Rhizobium and Azotobacter? That'd be actually a really good question. And that would bring in the nitrogen cycle along with DNA sequencing, along with the classification. Anyway, who knows? Who knows what they're going to do? Right. Let's look at biotechnology because I do have a strong feeling that that potentially is coming. Let me just move that down slightly. Imogen, the other thing to note is if you are looking for something specific, my videos are in the playlist specific to the topic area. So if you go to ecosystems playlist, you'll see the content on nitrogen cycle specifically. Okay, so asepsis then, if we are going to culture bacteria, if we are wanting to do any form of investigation, ideally we want to make sure that there is a sterile environment. We want to prevent contamination. We know that pathogens can be airborne. We know that spores are potentially going to be airborne. So therefore, if we want to ensure our investigation, whatever investigation we are doing, if we want to ensure that it is only looking at the organism or the material that we're interested in, we want to ensure that we have asepsis. So we want to ensure we have a sterile environment. We want to prevent contamination, and we also want to use what's known as aseptic techniques. Now, these are these practical measures, and this is, so someone said about the PAGs. So this is your PAG, I want to say it's either PAG 7 or PAG 9, I can't remember off the top of my head. That is specifically where you will have looked at using culturing bacteria. You will have potentially used antibiotics or antiseptic solutions, and you have been looking at maybe the zone of inhibition, let's say. You should have done this where you would have inoculated, and what that is doing is we're going to introduce microorganisms. Now, those microorganisms could be bacteria or it could be fungi. It really depends on the question. It depends where they want to go with this. So if we're going to use inoculation, we are going to introduce microorganisms into a culture medium, and we should know that we are going to potentially use an inoculating loop. And if we're going to use an inoculating loop, we're going to flame the loop. By flaming the loop, what we are going to do is we are going to turn the Bunsen onto the roaring hot flame. So it actually shouldn't be an orange safety flame. It should be a roaring flame, which is a blue flame, which is not really visible apart from what we call the cold triangle. So the reason we've got a hot roaring flame is we are opening it to allow lots of oxygen in. Therefore, we have got a blue flame that is really, really hot, and we're going to make our inoculating loop or spreader, depending on what they've given you. We're going to make sure that that is really red hot because we want to kill the bacteria that are already on in the inoculating loop, the metal. If it is a spreader, we're going to potentially use a glass spreader. So we're going to make it hot enough to kill anything that was there, but not too hot to melt whatever you're using. So if it is a plastic spreader, you're not going to use a Bunsen. So again, it's really important that you're looking at what have they given me in the question. If I'm using a plastic spreader, then I'm going to need to use a disinfectant. I'm going to put it into a disinfectant solution. So this is where students get caught out because they're like, oh yes, I'm going to talk about flaming it, when actually the question was giving you something which was plastic. So you have to adapt to the question that's in front of you. If it's a metal inoculating loop, then of course we're going to flame it. If it is a plastic inoculating loop or spreader, then we're going to put it into a disinfectant. So again, make sure you are using the question in hand. We are also going to want to flame the tube that we're using. So we have a specimen tube, which is normally made of glass. We're going to flame that. Now, when we actually use a Bunsen in a room, what that will do is that will create a convection current. So the hot air rises. This is where your GCSE comes in. Hot air rises because the air particles are more spread out. They're less dense. They rise up. That is then going to move outwards because of the change in air pressure. So we end up with a convection current that's happening around a flame. Now, by doing that, we're spreading out any of the air particles that could have contained those pathogens, that could have contained those contamination, those other microorganisms that we don't want to have there. So we're wanting it to spread out. We're wanting it to ensure that it's not settling. If it's on our tube, we don't want it to settle there. We obviously want to kill any bacteria that might be on the neck of the tube. We want to only transfer the sample that we want that is in our pure culture. So we're going to make sure we are working in a sterilized area. So for those of you who are considering going into a lab, a lot of the times you'll see a lot of these videos, which are really good videos out there showing you the day in the life of someone doing these actual experiments. They are in potentially a fume cupboard. So you know in school you will have used a fume cupboard for chemistry. If you're lucky enough for biology, you will have used the fume cupboard, but you may have had to have moved into a special lab that's got it. That is important because what the fume cupboard does is the fume cupboard is going to actually suck out the air. And if it's sucking out the air, it's taking those contaminants out. So you're sterilizing the area, but you will also find that they will also have abundance in being used because that is obviously helping the area be sterilized. So if they're going to give you anything to do with the biotechnology side of things, they're obviously looking at your understanding of the population growth curve. So you are aware, it is, let me just remove this and this. It is important that you are aware of your population growth curve. It's important you know that obviously if we're going back up here, if we're going to be growing them, just let me add this in a second. We need to think about let's try that again. Liquid medium that we want to use. So if we're using a liquid medium, okay, if we're using a liquid medium, we are growing the bacteria in a conical flask, let's say, and we are giving them a liquid medium. If we're growing them on agar, then in a petri dish, then we are needing to make sure again that the agar has got the nutrients. So the medium that we are giving, we want these cells, we want these prokaryotic organisms or these fungal microorganisms, we want them to be able to go through the process of binary fission if we're talking about prokaryotes, or budding if we're talking about fungi. We know that they do budding, but they're going to do the process of mitosis. So we know we want them to have the right nutrients to be able to grow, and so they should follow what's known as your population growth curve. So if we're looking at a population growth curve of bacteria, they may change it and say it's fungi. It just depends in the question. We know that we're going to use a logarithmic scale. So we know we're using a logarithmic scale because, let's try that again. We know, remember what we said before, if we're doing binary fission this time, instead of us doing DNA replication, if we're doing binary fission, we know that our bacteria will multiply, they will replicate, and they will replicate, let's say, every 20 minutes or every 30 minutes, depending on what they say. So let's say they are going to replicate every 30 minutes. Well, you can quite clearly see we've got 7 hours. So if I've got 7 hours, I would do 1 times 2 to the power of how many 30 minutes go into 7? Well, we know that there's 14 because there's 2 30 minutes are going to be 1 hour. I've got 7 hours, so I therefore have 2 times my 7, which equals 14. So my generation size, if I can correct that, let's try that again. My generation size is 1 times 2 to the 14. So if I were to work that out, what would I get? If you're still here doing your math, can you do that for me now? Or I can get my calculator up. 1 second. Where are you? There we go. So what have we got? 1 times 2 to the power of 14. I therefore would get 16,384 if I were doing it for that long. Now, if I want to actually represent that in a logarithmic scale, you should know how to use your calculator. So a log 10 in the bracket should be 16,384, because I feel like they're going to make you do that. And that would therefore give you, oh, I just shut my calculator down. Didn't mean to do that. Hold on. Calculator. Still there. Let's go. Right, where are you? Log 10. 4.21. So therefore that would equal 4.21. So then I would go to my scale over here, wherever 4.21 is. Let's say it's there, and that would be where I'd be plotting my population for my 7 hours. So it really depends how they're going to ask you to do this. They may ask you to read the logarithmic scale. They may ask you to actually do some calculations. It just depends. They may ask you to read off a graph. So we know we use a logarithmic scale because it allows us to see the difference in the numbers because they'd be too great otherwise. So if we had, for example, 2 hours, we would want to be showing 1 times 10 to the 1 times 10, 1 times 2. Why isn't it log 2 as they're doubling? No, multiplied by 10, because whenever we tend to use the logarithmic scale in biology, especially for OCRA, they use the logarithmic scale to 10, and so that's why it's log to 10. So if they're going to give you a log to 2, they will decide to do that. It will be very obvious, but I'd be surprised if they do that because OCR loves a log 10. So that would be more that they're going to use that. It doesn't mean they're not, just to be clear. So it's important to look at the question and the scale on here. So if you're looking at the axes, look at what they've given you. But in biology with OCRA, they tend to use the log 10. That would be what I would say, but be aware, because if they do it as a log 2, crumbs. See, this is where I'm trying really hard to learn how to write, but it's not quite writing in the right way. If they do a log 2 there, it will say it's log 2, but they tend to do log 10. So therefore, you should know how to use your calculator for that. Remember, we're not assessing we are assessing your math skills, but in biology. And so the exam board will have decided what they're going to use. But you're right, it's important to look at what they've given you in the question. But with OCRA, a lot of their questions are log 10. So do make sure you're using the right bit of your calculator on that one. And therefore, that's why I wrote log 10. And you'll note it in the question as well. Give me 1 second. Let me just double check that, because they gave a graph last year. Let me double check the graph last year and what they actually put on the scale. No, I don't want that. Sorry guys, give me 1 second whilst I quickly find this. Yeah, on the scale in last year's paper 1, it was a multiple choice question. They actually wrote on the scale log 10. So hopefully that helps you to know. Why do we times by 2? We're timesing by 2 to the power of, because we're multiplying every time. So if we're doing binary fission, we go from 1 cell to be 2. If we're doing mitosis, we start with 1 cell. Through the process of mitosis, we get 2 daughter cells. So we're always doubling it because of the fact that we are either doing binary fission or we're doing mitotic division. Hopefully that helps you. But it is important, and it's a good question. These are why it's good for you to ask these questions, because what if they do decide this year to go, we're going to assess your ability to read the scale. So it is important to double check that one. Could it be done for meiosis? I don't think they would use it for meiosis, because every time we're doing a reduction division, and we would be, but yeah, I mean, potentially, if they were going to look at it with regards to how many cells would you get. Oh, I don't know on that one. I'm not sure. I would say probably not. Right, let's just keep going on this one, because I really want to get the biotechnology part of the topic done. Yes, it is the 2n formula. You're absolutely right. We have to work backwards from a logarithmic scale. Yes, you could You could be asked to work backwards, absolutely, which if you are, if you know, so let's say I gave you the thing that was we worked out to be 4.21. Let's say we had it for 5.75. If you read off the graph and it was 5.75, wow, that is so bad. Oh, it's embarrassing that I'm doing so bad. I'm getting so frustrated with it, but I do things so quickly. My writing's bad as it is, but doing things so quickly makes it even worse. If they were to ask you to read off a population growth curve, and let's say we've read our log 10 scale to be 5.75, then we would need to, on our calculator, do 10 to the power of 5.75. And that would give us working backwards of how many were there. I do feel like something like that is coming up. I'm not sure how, if it's going to be about working back from a serial dilution or if it's going to be reading from a graph, but something like that. I do have a strong feeling that that is going to come through. We were doing a lot of practice on that with the course. Looking at a growth curve, obviously you should know about your lag, you should know about your log, you should know about your stationary, you should know about your death phase. Now, be aware the lag phase is where there is, to start with, no bacteria dividing. So do make sure if you are reading a graph that you're looking at the scale, where is that lag phase? The lag phase is where there's no bacteria dividing. So you need to be looking at the scale. Obviously, I haven't got a so that you can look at the scale. So let's say in the first half hour, in this case, in mine, actually, that's not true. It's not a half hour because, oh no, let me just, hold on. There we go. If I do it, if I go too far, oh, no, did it again. Oh, you're seeing me like fail like straight away, but that's okay. It's okay to fail because I'm learning as I do it. So if I go and I need to be specific to the point at which it is not, oh, no. Actually working, so let me rub that out. Try that again. So there, and then if I draw this at that point there, whatever time that is, and that's less than half an hour, that is where there is no growth that is actually happening. Or is it always the maximum value on the x-axis? It really depends on the graph that they give you as to what you're going to read and what they're going to give you, but knowing to use that scale is really important. So no bacteria growing, they're making enzymes to deal with the food source. So let's say, because I do have a strong feeling about the lactoperon, let's say that we're going to give them lactose here. So if their food source is lactose, they therefore need to have the lactose binding to the repressor protein. They then need transcription, translation of the lac A, lac Y, lac A, lac Y, lac Z. And so therefore that takes time. So this is where it could link together. Once those enzymes have been produced, once they are able to go through the mitotic division, no binary fission, because we're talking about prokaryotes. So do be careful you don't say the wrong thing. Then we go and into this log phase. This is the exponential growth. Again, be aware they could ask you to identify where that is in the graph. So the exponential growth is where you have that constant rate. So it's better to say, it's not better to say log or exponential, it's either or. If they want you to name it, you would say the lag, the log, the stationary, the death. But if you described it as a exponential growth, that's fine as well. I know this exam feels like there's so much to learn, and there is, and it's hard, but this is the reason you've got these videos, that I'm doing these for you, so you can go back and watch it again. If you're finding that this is too much, don't watch this live. Go and watch my smaller videos just on those key topic areas. And that will help you. Just having a little bit of a recap of it will help you prepare for the potential of what's coming in Friday's exam. There is a lot to learn, and I know that. So I feel for you, but be aware that you still have the time to do the revision, okay? You still have this time. Okay, stationary phase. What have we got the stationary phase? Stationary phase, we've got the cells are dying, but they're dying at the same rate of being reproduced. So be aware that if we're looking at a log phase, so if we had, let's say that's 8, I'm going to give a different number now. That's 8, and down here is 5. Oh, Water, thank you so much. And thank you if you have purchased my revision guides. That's really helpful, because that's basically how I earn my money through the revision guides. And these are the resources that I have got. Oh, thank you. Yeah, I've tried my best. Obviously, I've got my private clients who help me make a living. Not nearly enough of a living as I'd like to make, but it means I get to spend more time with my family, so I do appreciate it. But I know that not everybody can afford tutoring, and also right now the stress and the pressure you feel under, sometimes just having something extra just to consolidate that knowledge, or just to answer that one question that was like, oh, what is it? I just don't understand. It could be the difference of 1 mark that gets you into a grade boundary. Where do you purchase them from? If you go to my website, which is UK Biotutor, hold on, I'll write it here. UK Biotutor, biotutor.com. If you go there, that is where all my revision guides are. There's also a my 4.5-hour course that I did in the Easter course. There are pre-recorded videos there as well. Oh, thank you. I really appreciate it, guys, because sometimes even I can feel disheartened with the work that I'm doing and people not actually engaging. I felt so funny yesterday when I had I think there was about 10 people in the live. I was really disheartened. I was like, oh, and I know it was during the chemistry exam, and right now it's during the maths exam, but I'm trying to make sure that these resources are available for anyone. It doesn't matter what exam you're doing, and it doesn't matter what level you're aiming at. I've tried to make them for those of you who are aiming for the C, and for those of you who are aiming from the A star. All of it is accessible as best I can. So I have been working really hard to build them. So I do appreciate and thank you for purchasing them. It does make a big difference. It means I can do these sorts of things that I love to do. I want to help. Anyway, I need to get back. My ADHD means that I go off on a tangent, but I can always bring it back. So this is the key thing. Right, back to I was talking about the death phase, and one of the questions they set out in the past was about where is the highest death phase? No, that wasn't the question. Where was the highest number of cells dying? And students were given, it was a multiple choice question, but what they can do is take those multiple choice questions and switch them to being a long response question where you're having to explain what's going on. So I always try and show you what's going on. So if I have log 8, so I've got log 10 equals 8. So therefore, oh my gosh, let's try that again. My writing, so hard. Log 10 equals 8. So therefore, I know I need to do 10 to the power of 8 equals whatever number is there. So if I do that on my calculator, or if someone else does it quickly for me. So if I have 10 to the power of 8, I should be able to do that, and yet I can't. Why can I not do that? Because my brain is shutting down. Obviously, that is 1.0 times 10 to the 8. That's crazy that I just need to, that's it in standard form. Anyway, 1.0 times 10 to the 8. If I were looking at the log 5, let's say actually the number that are dying could potentially be just 10 to the 3. And if it's 10 to the 3 that are dying, there is less dying than there were at the stationary phase, is what I'm trying to say. So because this is a logarithmic scale, you have to know that at the stationary phase, the number that are being produced are dying at the same rate as the number that are the number that are being produced, the number are dying at the same rate, which is keeping the population size at a constant level. So therefore, that number that are dying are actually higher than those in the death phase, which seems bizarre. But what we're saying when we get to the death phase, which is important, is that the cells are dividing at a at a faster rate than they are dividing. So it's one of the ones in which they try to trip you up, where they said where was there a higher death phase, and it was like A, B, C. Why was there a higher death rate? A, B, C, or D? And obviously a lot of students went, oh, it must be D, and it wasn't. It was actually at C, because C was much higher. So therefore the rate of death is faster. There's a lot more there. Oh, thank you. Someone's just bought one of my study guides. Thank you so much. I got a little notification. Thank you. I appreciate that. Okay, what is actually going to cause that stationary phase and then to the death phase? Now, that is limiting factors. What are those limiting factors? The nutrients available, the oxygen levels, the temperature, the buildup of waste, the change in pH. So make sure whenever you are answering a question, yes, you've got your list of limiting factors, but you need to relate it to the specific question that they have given you in the exam. So whatever that is, make sure that you are talking about it. So here we have glucose. So if glucose is my respiratory substrate of choice, I would expect my biomass to increase because my glucose has decreased. So my nutrient, my substrate, has decreased. Yes, space would be one, because as the population is growing, the space available will decrease. Absolutely. I'm just looking through it right now. It's so detailed. Thank you. Oh, thank you. I really appreciate that. They are very detailed, and the diagrams are all my diagrams as well. They are ones that I have been learning to make on Adobe Illustrator. All of this is just me. I am me doing this YouTube channel, doing my tutoring. Just it's crazy. I've learned a lot in the last 2 years, and I hopefully will keep learning more to keep improving. So thank you. I do appreciate it. So glucose is decreasing, the biomass is increasing. I do have a feeling that biomass is coming in, whether it's about energy transfer or whether it is about biotechnology. I actually feel like all of that is really coming through. So we have this increase in the biomass. We've got this decrease in the nutrient. And if we are producing, I say we, if the microorganism is producing a primary metabolite, it will be one of those things where it's needed for normal functioning. So obviously we know that through the process of glycolysis, glucose is broken down into pyruvate. We know that that pyruvate is then going to go into the link reaction. And we know that then we have our Krebs cycle. We know we have our oxidative phosphorylation. So if we have a primary metabolite, we have a substance which is being formed as an essential part of the normal functioning of that microorganism. I have a question. If aseptic conditions aren't carried out properly, can other microorganisms come in and increase? Yes, that's exactly the kind of question where they could ask you as to why might the population growth curve not follow the expected trend. And it could be that in the practical situation, the person who was doing it didn't account or didn't use the correct aseptic conditions, and therefore there was competition. How would you know that? Where there could be a extra bit of growth on their plate, for example. But yeah, that's absolutely a great type of question, and them looking at understanding that there would be competition and how you would have the expected population but actual viable population as well. They've sometimes done that. Now when we're looking at primary metabolites, the primary metabolite is usually formed during the active growth period. That's normally found in your log phase on the graph. If we're looking at a secondary metabolite this time, we're looking at something that's not essential for normal growth. That would be used by the cell to, for example, reduce that competition. Interspecific competition, we therefore would have these secondary metabolites, and those secondary metabolites are normally formed during the stationary phase. So if they're going to ask questions on that, they could be looking at, and again, this is something I do feel is coming your way, but again, I could be wrong. I feel like it's coming your way with regards to fermentation. So biotechnology, if we're going to grow these microorganisms, we're going to need to use our aseptic technique. We're going to need to have that inoculation happening. So if we're using batch fermentation, we've got our microorganisms, which are going to be inoculated. We're going to be transferring them, and we're going to do that in a fixed volume of medium. So remember that medium that we're giving them. Oh, no, let's try that again. So the medium that we're giving them has to have the right nutrients so that they can thrive. Because if we don't give them the right nutrients, they're not going to be able to thrive. So it could be that they give you a question where they've been trying to grow the microorganisms in different nutrients and seeing the effect that has on the population growth curve. So looking at the biomass. There's a little squirrel. Sorry, I'm being distracted here. Oh, I'm sorry. The ads that you're getting, when they put the ads in there, the ads are the only way in which I can actually earn any money from my YouTube channel. So I do apologize that that is happening to you right now. But they should hopefully be skippable ads. So I do apologize, but you will be able to hear. So intra, yes, is, no. Intra is within species. Inter is between species. So how I remember it, just to answer that question, intra is the same. Inter, between. Oh, I put an extra, looked like an extra E there. So inter has an E, between has E's, ignore the E and same. Intra is same, okay? And so we have to inoculate it. We have to give them the right nutrients. So I feel like they may give you a question where they've given those different nutrients, whether it's lactose, maltose, sucrose, fructose, glucose, depending on what it is they want to ask the question on. So if it's a batch fermentation, it's in a bioreactor. If it's continuous fermentation, we're going to also... Oh, thank you. Only 5 to 10 seconds max. Yeah, but it does help me. I mean, I managed to earn £2.97, I think it was yesterday. That £2.97 means that I can work for these 2 hours that I've given you today for £2.97. Woohoo! But it does actually make a big difference. It's crazy. The little bits of money that come in are all added together, which allows me to be able to do these things for you. So I'm sorry it's a nuisance. I apologize, but it is my only way of actually earning during these free sessions that I offer. So yeah, I appreciate. Is the diagram apparatus likely to come up as I saw in a previous paper? It could come up. It could come up because they've already asked you to plan an investigation. I don't feel like they're going to ask you to plan the investigation again in paper 2 or paper 3, but I do feel like they're either going to get you to look at something and talk about why you would use them. So giving you a picture that looks like this, which they have done in the past, and explaining why you've got all the different things that are needed. Or it's going to be a case of they give you a student did this, improve their investigation. So that's the sort of thing that could come up. I do still feel a statistical test is coming up. Yes, if it's not, I know it hasn't come up in paper 1. So I do feel like a statistical test is coming in paper 2 or paper 3. And this is the thing you have to remember. If it doesn't come up in paper 2, you've still got one more paper that it can come up in. It just depends where they put it. Chi-squared would be more likely in paper 2 because of the genetic side of things, and those go hand in hand. But I don't know. I still feel like a Spearman's rank is coming through, and I feel like that or a t-test, so it could come up in paper 3. I don't know. Okay, back to this batch fermentation during the stationary phase. Okay, now remember that stationary phase, the microorganisms are going to carry out the biochemical changes, and those biochemical changes are going to produce, give me a second, I need to just make this a bit smaller. Biochemical changes are going to produce what's known as those secondary metabolites. So if you are looking at the population growth curve and you see that they have stopped it at the stationary phase, you therefore know that you are trying to get the desired product, which is a secondary metabolite. Yes, the chi-squared one is the observed and the expected. You're given all of the statistical test equations in your paper. I am going to try, I won't be able to do it today. Actually, I might be able to go live tonight at 9 o'clock again, depending on how tired I'm feeling. I'll try to go live at 9 o'clock, and whatever I haven't covered, I will come in. Yes, even standard deviation, they will give you that equation. If you are wanting to do speciation though, do go and check out my video on it. I think everybody feels underprepared. Paper 1 was interesting. Let's just put it that way. I feel like you've got a lot of hard ones coming. Is secondary metabolite only for continuous, only for batch? No, it really depends. I mean, it depends how long they leave it before they're going to remove the product. So it's important to note, if they give you a graph, you're looking at the population growth curve. So you're looking at the biomass that you're getting. And if you get that stationary phase and they've given you a graph and they're saying that they're removing the product at a point. So hold on, let me show you what I mean by that. Oh, yep. So let me just finish this quick one off. So obviously, stocked before the death phase, products are harvest, system re-sterilized, and then you're going to repeat it. You're going to have this continually adding of the nutrient agar. For those of you who are saying, are there any specific papers I would recommend doing? Yes, 2019, 2021, and 2022 would be my suggestion. Starting at 2019 would be where I would go. I'd also suggest 2019 AS paper, paper 2, I believe. Is it paper 2 or paper? Oh, I can't remember if it's paper 2 or paper 1. Actually, it might be paper 1, which has the, oh my gosh, it has the Scottish Wildcat question in it. Are batch fermentation and continuous fermentation both for the agar gel method or no? It depends, because you got to be careful. What you might do is in the lab, you might do your liquid broth or your agar plate so that you can grow the bacteria. It may be even, and I'll show you in a minute, it may be that you are going to genetically modify the bacterium to insert a gene of desire, and then you're going to culture it, and then you're going to transfer that into a batch bioreactor, so batch fermentation in the bioreactor, or you're going to transfer it into a bioreactor which is for continuous fermentation. It really just depends. So it could be asked on either front. It could be asked on the sorry, I shouldn't laugh. This squirrel keeps coming up to my up on my fence. It's getting so close and then just staring at me, which is really funny. Hold on, let me see if I can show for those of you on my, oh, I've just liked my own thing. I didn't mean to do that. Oh, this is where I'm like, how do I flip my camera? Oh no, you can't see it. Oh, crumbs. Oh no, because I've got my background. I can't remember how to do this. Oh, I'm just going to flip it back. Sorry. Doesn't work. But it's really funny because the squirrel, I'll try and actually, maybe I can put a picture up on here because I took a picture yesterday. It keeps coming and here we go. Coming and staring at me. Hold on. I'm going to put it in my slide here. There we go. This little guy keeps coming and staring at me here, which is very distracting. This is the thing, when you're doing biology, when you're doing your study and your revision, you guys are like, shut up, Christine, get on with it. We're doing our exams and you're talking about squirrels. But this is the thing that happens when you've got ADHD. You have, unfortunately, these distractions, and it's okay to have the distraction, but the key thing is having the skills to bring yourself back to where you are and what you were talking about. So yeah, this little guy keeps coming and saying hello and running off. He's getting so brave as well. Right, so what I was talking about is if you are given a graph and it is a population growth curve. So here we go. I've been told that I have got my glucose has decreased. I've got my biomass as it was going up. And we've got time down here. And what they've said is we are going to extract so we're doing a continual fermentation and we are going to extract at this, point here. And if I'm extracting at this point here, if that's where I'm going to extract, then I know I'm at the stationary phase. So it's important that you are aware on the graph, what have they told you? If they are extracting at a point where we have a stationary phase, then we know that we have got a secondary metabolite. If we are extracting and they tell me in the graph that we're going to extract at this point here, then we can assume that we have got a primary. Oh my gosh. It really does not like my writing. Try that again. Primary metabolite. So it really depends on what they give you in the picture and in the graph. So if we come back to what we were talking about before, so stationary phase is a point where there are the highest death rate, but it's equivalent to the birth rate. Absolutely. So if we're looking at this one, the key thing to note is that we are talking about continually adding in ammonia and air, continually adding in glucose. So why are we giving it the glucose? Well, the glucose we know is for respiration. I know everybody's like, please, no more respiration. I have to say, I was a bit disappointed in the paper when I saw it. I feel like the questions weren't as challenging. It sounds ridiculous. They were challenging, but they weren't dissimilar to previous exams, and I'm a bit disappointed in the person who wrote it for that. Anyway, respiration for glucose, ammonia for either, and this is the thing, most of you might go, well, the ammonia is needed for amino acids. Oh my gosh, I'm gonna try that again. Nope. What time am I at? Oh, I need to finish soon because I've got a student coming at 12:30. I'll do a little bit more to finish this off, and then amino acids. So most of you tend to go, oh yeah, it's about amino acids, or you tend to go that it's about proteins, but they could actually ask you about nucleotides. So they could ask you about RNA nucleotides. They could ask you about DNA nucleotides. Patterns of inheritance could come up, and I did all that yesterday. So if you haven't seen my live that I did, if you're in your chemistry exam, I did a 2-hour live yesterday on a lot of the patterns of inheritance topic area and the gene control topic area as well. So do be aware, the ammonia and the air going in, obviously oxygen going in if we are doing respiration, but they could bring in the nitrogen cycle here and they could talk about the air and it containing nitrogen, because they could actually talk about you culturing a oh my gosh, nitrificating bacteria. I couldn't think of the word there. Nitrificating bacteria. So they may ask you about, oh my gosh, no, that's not what I meant. I didn't mean that. I'm thinking of a nitrogen fixing. I knew, my brain just stopped there. It's because I've been going for too long. Nitrogen fixing bacteria. Nitrogen. Oh my gosh. Gosh. Let's try that again. Oh, I wonder if I can. Nitrogen fixing bacteria. I do feel like they could bring these two together. Bacteria. So they could make a question. They could make a question which is about how you would produce let's say a fertilizer for soils. And so if you were going to produce a fertilizer for soil, then you would want to have nitrate ions. How are you going to produce the nitrate ions? Well, if you use nitrogen fixing bacteria, you could do nitrogen fixing bacteria to produce the ammonia. The ammonia could then be used to be converted into the nitrites and nitrates, depending on which one you've got. So the nitrogen gas could be used for the nitrogen fixing bacteria, and therefore there wouldn't be ammonia in your source. There would be the nitrogen so that they can do nitrogen fixation, because you're trying to get the ammonia. The ammonia, if you're trying to get it for that conversion into, for example, nitrites, into nitrate, so that we could also bring these into it if we want to get nitrates. So it really just depends where they want to go with the question. So yeah, it really does depend on that one. The other thing just to note before I finish, and I am going to have to finish soon, is they could talk about a zero dilution. So they could talk about zero dilution. They could talk about how do you do a tenfold dilution. So if you are doing a tenfold dilution, and let's say we've got 2%, we should know that we are obviously going to bring it down. We're doing a tenth, and therefore we have taken 1 centimeter cubed. So 1 divided by 10, which is my fixed volume. 1 plus 9 is 10. So I've got 1 over 10, which gives me 0.1. And if my concentration was 2, I would have 2 times 0.1, which equals 0.2. And if I keep doing it, each time I am diluting it by a factor of 10, I have got a 10 to the minus 5. Now, don't get caught out on the question if they were to ask about if you are going to plate it and you are going to take 0.1, or sometimes they put 0.01. So if I'm going down to the plate and I've taken 0.1, then I need to be aware that that is obviously, if I had 10 centimeters cubed in here, then I need to dilute it down even further because I'm only taking 0.1. So if I've got 10, how do I get to 0.1? To get to 0.1, I would divide by 100. If I am trying to get to 0.01, I would need to divide by 1,000. So it depends on that question, what they're asking, where you need to go. I'll do a little bit more on that later on, because I do feel like a zero dilution question could come up there. But do be aware of that in case it does. The other thing is, as I was showing before, if they give you a logarithmic scale, they ask questions such as why they shouldn't use the logarithmic scale. Obviously, it's about that different numbers. Always give 2 quoted numbers, and they could give you the total number or the viable number. Why might there not be the same number as the total number that could be sustained, and yet they're the ones that are viable because some are dying. And the only other thing they could bring in, linking to biotechnology and linking to manipulating genomes, is the thing where it says about if I have a plasmid and I use my restriction endonucleases, or if I want to isolate the gene using my restriction endonucleases, or if I am going to, hold on one second, just move that so you can actually see what it says. So I am going to have my plasmid taken from my prokaryotic cell because it's a good vector. I'm going to isolate my gene, or I'm going to have mRNA. Now the mRNA, if I'm going to treat it with reverse transcriptase, I'm going to use a enzyme where I started off with a single strand of mRNA. I need to isolate that mRNA. And then what I'm going to do is I'm going to use reverse transcriptase. And what that will do is that will therefore allow me to produce a double-stranded DNA, which is complementary DNA. And then what I'm going to do is I am going to use restriction endonucleases to cut apart so that I have the specific base sequences, so that they have what we talked about earlier, the sticky ends. Oh my gosh. So frustrating. Let's try that again. Sticky ends. Seriously, how do you guys write on this without it being shocking? So I've got the sticky ends. I'm then going to have my DNA ligase, which is going to stick them together. Now, the key thing to know is the DNA ligase is going to stick them together. The sticky ends is about the complementary base pairing. Well, sometimes they won't stick together in the right way because actually the plasmid may come back on its own. And so therefore make sure that you are looking at what they've told you in the question. We're going to have the recombinated plasmid introduced to the prokaryotic cell, and we do that through either we could use a calcium rich solution and increase the temperature, give them a heat shock, or we could do electroporation, or we could do electrofusion. It depends on the question, what they're asking. So be aware, it may give you a different example. We're then going to grow them, and when we grow them, we are going to grow them on an agar plate. And if we're growing them on an agar plate, hold on one second. If we grow them on that agar plate, so we're going to grow them first, we're going to use marker genes so that we can identify the ones that we want to inoculate. So obviously we may get growth of ones which are not fluorescing. We only want to grow those that are fluorescing. They could be the marker genes. We want to grow them on antibiotic agar. The antibiotic agar therefore will kill those that don't have the antibiotic resistant gene. And then once we have cultured them, then we transfer them into the bioreactor. So we need to grow them on an antibiotic medium. We need to have a marker gene that fluoresces. So we need to expose them to, for example, UV light so that we can see them. And then we're going to transfer them. We're going to inoculate that bioreactor. We want them to multiply in the fermenter. And if we're looking at the production of insulin, for example, we would have genetically modified bacteria, which are going to produce the insulin through the process of transcription translation. We're removing the carbon dioxide because we don't want the acidity to change. We don't want the buildup of waste material. We're going to remove the product, but we're also going to remove cells that could be dying and cells that could be living. We need to ensure that we control the temperature. So we've got this water cooling system and we've got the nutrients going in. It really depends where they want to go. We obviously need to separate them. We need to purify it if we're going to insert it into humans. Now, they have done a question in the past, which was an AS question, which was looking at a genetically modified synthetic insulin. And so it was an AS paper. I can't remember exactly which year it was, but it was about the genetic modification of this insulin molecule. And this is where they could take an AS concept and add it into how we could then use that in a bioreactor to pull the information out. So be aware that the content that's covered in AS papers, sometimes they throw in little bits of A2. So the 2019 paper is a really good paper if you're looking at AS, if you have the time, that I would suggest doing. So yeah, that would what I would suggest, guys. I am going to have to finish because I've got a client at 12:30, and I've also got to prep for my group session tonight. So do I have any specific module 4 that would come up? I've covered most of it here, but I would say definitely go over the immune system side of things. The whole course is in my videos and playlist linked to the module specifications. So definitely go and check those ones out. I will try my best to come live at 9 o'clock tonight. Because I cannot be live tomorrow, I'm afraid, guys, until the evening, tomorrow night. So I will try and go live at 9 o'clock tonight. And then I will go live tomorrow, probably at 9 o'clock as well, if I can. Obviously, I've done 2 previous videos covering preparation for paper 2. So do check out my last 2 videos that I did as well. I will hopefully see you all later. Thank you for those of you who have been here with me and who have responded, shared, etc. my resources. I do really appreciate it. And thank you if you have purchased my study guides. I appreciate that as well. I have done there's a study guide for manipulating genome. There's a study guide for cellular control. There isn't a study guide for biotechnology, I'm afraid, because I haven't got around to doing that one yet, even though this is the one I feel it's coming up. But I didn't get a chance to do that yet, which is why I've done a lot of work on this one. You are very, very welcome, guys. You're very welcome. Good luck. Keep going. And I will see you hopefully tonight at 9 o'clock. Do keep a watch out. And I will do a session tomorrow before paper 2 on Friday, and you'll be sitting your paper 2 whilst I'll be in Pilates. So guys, have a good one, and I will see you later.